Hi Tiruvira, very good evening. Very good evening, Shrikant. After a long time, I'm seeing your name. Happy evening. How are you? Yes, Nabita. Very good evening. Hi, Nabita. Hi, Ahmed. Very good evening. Happy to see all your names. Okay. So today we are going to discuss, of course, the cell, the unit of life. The second part. Yes. Are you all ready? So before we proceed to the topic, let me give a small game, okay? Can you identify what is this organelle? Hi Sanjay, very good evening. Very good evening Sanjay. Yes Nabita. See there is a small puzzle here in the screen. I hope you can see all of you. I gave two pictures. Can you guess what is this actually? Let me see who is telling me first. Yes, 5, 4, 3, 2. Yes, my dear children, Navita, Sanjay, Tarunika. I gave two pictures. You could see a power station. I hope you can see a cell also. Yes, can you guess? Very good, Sanjay. Excellent, excellent. It is mitochondria. Very correct. Excellent answering by Sanjay. 100% you are correct. Yes, Dharini. That is the correct answer. Very good evening. Yes, Shrikant. Very correct answer. Excellent. Okay. A small funny line for you all related to mitochondria. See, a rowdy got after doing some illegal activity and he went to the cell. He was caught by police and he was arrested and he is into the cell. Now in cell, he changed his name as mitochondria. Can you guess why? So he did some illegal activity. He was caught by police and he changed the name as soon as he entered into the cell as mitochondria. Can you guess why? Yes, Nabita. Yes, Sanjay, come on. Shrikant. Yeah, you're fine. Why that fellow wants to change his name as mitochondria? Can I reveal? Yes, 5, 4, 3, 2. Yes, come on, Sanjay. He generate power. Is it so? No, he wanted to be powerhouse of the cell. Yes, thank you, Shrikant, for your words. Thank you so much. They are all supporting me, Shrikant. Yes. So you can even inform all your friend children to join us. Anyway, let us continue now. So we are going to discuss endoplasmic reticulum. Cell wall we finished in the last class. Yes, almost reached. Yes, you're correct. Okay. So in endoplasmic reticulum, as you can see, who discovered it is Porter. Porter only discovered the endoplasmic reticulum in yes, you enjoyed my joke, Hagamer. Yes, Porter only discovered, of course, before that Garnier were also there in endoplasmic reticulum. As you can see, the endoplasmic reticulum is closer to nucleus. So it is closer to nucleus and you can find in the form of, yes, Jeevan, very good evening, Jeevan. Very good evening. We just started, Jeevan. You missed one funny joke. Okay. So it is closer to nucleus, of course, and you find the endoplasmic reticulum in three forms, either in the form of cisternae or in the form of vesicles or in the form of tubules. See, the cisternae and vesicles are closer to the nucleus, whereas the tubules are closer to the plasma membrane. And the cisternae and vesicle will have the ribosome along with that. So ribosome is present here, whereas here ribosome are absent. And we find the endoplasmic reticulum in all the cell organelles, all the cells except, can you tell me, where and all we won't find the endoplasmic reticulum? Yes, come on children. Yes, Ahmed, Sanjay. Yes, 5, 4, 3. Yes, Shrikant. 2 and 1. Yes, you won't find it in germinal epithelium. Yes or not? So you won't find it in germinal epithelium. Yes, Jayasurya. Vanakkam. Vanakkam, my dear Jayasurya. Very good evening. Vanakkam. Namaskaram. Namaskar. So we just started, Jayasurya. You missed one funny joke. Anyway, so endoplasmic reticulum, you may not find it in germinal epithelium as well as, of course, yes, you won't find it in prokaryotes and also in RBC. Yes, Mohammed, you are very correct. It won't be there in prokaryotes, bacteria. Yes, but we are discussing about eukaryotic cell. These two are lack of, yes, Sanjay, very correct, RBC. Is the correct answer. Excellent answer. Okay. So you won't find these two places in eukaryotic cells, endoplasmic reticulum. Yes, Nandika, thank you for your appreciation. Now let us differentiate smooth endoplasmic reticulum and rough endoplasmic reticulum. Can you tell me, children, which involved in protein synthesis? Which involved in protein synthesis? Is it RER or SER? Yes, Navita. Yes, Dharni, my dear. Sanjay. Yes, Shalini. Very good evening, Shalini. Very good evening. Just we started, Shalini. Cell organelle. We are into the differentiation. Yes, Vaishnavi. Very good evening. Very good evening. Yes, Tiruvira and Dharini. It is RER. Okay, it is the RER is the correct answer where we involved in protein synthesis. Yes, Tiruvira, yes, Dharani and Jeevan. Yes, Navita, very correct answer, excellent. Okay, RER as you have ribosome, it involved in protein synthesis. Okay. So can you give an example which organ has RER in its structure? Yes, Navita, it is rough endoplasmic reticulum. Very correct. Yes, very good evening, Vaishnavi. Can you tell me where do we find the smooth endoplasmic reticulum or rough endoplasmic reticulum? Rough endoplasmic reticulum we find in neural system. Can anybody guess neural system? Can anybody guess where it is found? Neuron. You remember, children? We have nasal granules. So this nasal granules, nasal granules will have the rough endoplasmic reticulum. Of course, it involved in protein synthesis. As you know, it is closer to the nucleus. Whereas smooth endoplasmic reticulum, devoid of ribosome, involved in lipid synthesis. And you know, children, this smooth endoplasmic reticulum. Yes, very correct. Jeevan, it is sarcoplasmic reticulum also. When you have endoplasmic reticulum in muscles. Very correct. Excellent. Yes, sarcoplasmic reticulum also we can say as an example. Excellent, Jeevan. Super. You are rocking today. It is correct. Sarcoplasmic reticulum is a part of endoplasmic reticulum where you find in the muscle. Okay. So here it is in the form of nasal granule. It involved in lipid synthesis. As you know, the questions will come like testosterone synthesis is under the control of which cell organelle? As you know, it is SCR or estrogen, progesterone, aldosterone. Okay. Apart from that, it also involved in detoxification. Okay. So detoxification is done by SCR as it removes some carcinogens also from our system. And apart from synthesis, it also transport the proteins. So transport of protein is performed by RER. Transport of lipid. Yes, Sanjay. Transport of lipid is done by SCR. Okay, children. Can you guess the picture now? Yeah. Can you guess the picture, children? Which organelle it is? You can see a truck. You can see proteins. Can you? Yes, can you guess, children? What does this picture indicate? The cell organelle. I have given you the truck for transportation. I have given you the protein also. Yes, Ahmed. Ahmed has given the answer. Yes, I am waiting for others too. Yes, can you all guess it? Yes. Yes, Amor. Yes, Vaishnavi. Navita. No, you already gave, Sanjay. It's correct. You didn't give, huh? Okay. Fine. Yes, Navita. Dharani. Shalini. Vaishnavi. Yes, it is transport. Of course, you are all correct. And I very well gave protein, my dear children. Transport of protein, who will do? It is RER. Okay. If it is packaging, like gift wrapper, if I gave and the protein, you can guess Golgi. But I gave only transportation. RER is the correct answer. Yes, Jeevan, excellent. Tiruvira and Ahmed, very first answered. Yes, Navita and Dharani. Yes, you are all rocking only. Anyway, it is transport of protein, little misunderstood. Good. Okay. Now we'll continue, of course. So as I said, transport of protein is by RER. Okay. Now we are going to discuss is about Golgi apparatus. So Golgi apparatus, of course, discovered by, can you guess who discovered Golgi apparatus? Can you guess, children? Who discovered Golgi apparatus? Yes, it is Camilo Golgi. Very correct answer. Excellent. Excellent, Sanjay and Dharini. Yes, Tiruvira. It is Camilo Golgi. Okay. And you know it is in 1898. He discovered it. It is named after him. Yes. Navita, Jayasurya, Sanjay, yes, Jeevan, Vaishnavi, excellent answering, 100%. You're all correct. You're all rocking today. Very good. It is Camilo Golgi in 1998. What are the other names of? Yes, Anishri, very good evening. So, what are the other names of Golgi apparatus? It is otherwise called packaging material. Any other name? The other name of Golgi apparatus is even called Dalton granules. Okay. Yes, Yogeshwari, very good evening. Hi. Yes, it is Dalton granules or Dalton complex. Very correct answer, Sanjay. Yes, good evening, Yogeshwari. So, yes, Parini, very good evening. Dalton granule is correct answer, Tiruvira. Excellent. Fine. So, you can find even Golgi apparatus in the form of cisternae, vesicles, and tubules. Yes, Priya. Very good evening, Priya. Happy to see you. We are discussing Golgi apparatus. So, cisternae, vesicles and tubules. Okay. Are there in Golgi apparatus? Of course. You can see the Golgi apparatus closer to the plasma membrane. And you can find a concave space and a convex trans space also. So trans space is there and cis space is there. Okay. So they are found closer to the plasma membrane. What is their main role? Of course, you know, whatever is synthesized, the raw materials will come to the Golgi apparatus. It will process and package. So it will package the protein and lipid. Okay. And you know, this is also involved in cell plate formation. Cell plate formation is under the influence of Golgi apparatus. Can anybody guess what structure of male gamete is contributed by Golgi? The structure of male gamete contributed by Golgi is dash. Which structure of male gamete is contributed? Yes, it received protein and lipid from endoplasmic reticulum is very correct answer, Ahmed. Excellent. 100% you are correct. Now can you guess which structure of male gamete, that is sperm, is contributed by Golgi apparatus? Yes, your time starts. 5, 4, 3, 2, yes. Yes, my dear children. Harinishri, Dharini, Yogeshwari, Sanjay. Any guess from any of you? The structure of male gamete contributed by Golgi apparatus is acrosome. So acrosome is contributed by Golgi apparatus. It also forms yolk synthesis, vitellogenesis. So vitellogenesis is by Golgi apparatus. And it involved in GERL. Yes, acrosome is the correct answer. Ahmed, excellent. It is acrosome. 100% you are very correct. You are rocking today. Yes, 8%, Jayasurya, very correct. Acrosome. Yes, Anishri. Yes, Jeevan, Yogeshwari. Very correct. 100%. You are all correct. Excellent answering. Now, what is the expansion of GERL system? Can anybody expand the GERL system? Expansion of GERL system. Yes, yes, excellent, Sanjay. Very correct. Excellent. It is Golgi body, derived endoplasmic reticulum, and lysosome. So these two will join and form the lysosome. That is the correct answer. Excellent. And Golgi body will be more in secretory cells. Like if you take pancreas. Okay. So it will be more in pancreas and liver, all these. Of course, it won't be found in RBC, non-secretory cells and all, it will be very less. Yes, Sanjay, Tiruvira, Navita, and yes, Agent Red, new to watch. I am welcoming you for the class. Very good evening. You can even give your name to make a call. Yes, Anishri, very correct. Excellent, all of you. Keep rocking. Okay, so these are the functions of Golgi apparatus. Okay. Now, next one we are going to discuss is about lysosome. So, who discovered lysosome? It is Christian de Dieu. It's a person who invented lysosome, of course. So, what is the other name of lysosome? What is the common name of lysosome? Common name. What is the common name of lysosome? Very good. Excellent, Jeevan. It's your day. You're rocking today, man. Jeevan? Yes. It is suicidal bag. Okay. So it is called suicidal bag. Common name. Okay. From where it is formed? We know it is formed from GERL system. Yes, Ahmed, it is suicidal bag. Tiruvira, Arinishri, Yogeshwari, excellent answering. Super. That is the correct answer. Excellent. You're all rocking today. So it is derived from GRL system, of course. Okay. So from the Golgi apparatus, assume this is the cisternae. If it is pinched off and form a vesicle, and that is what the lysosome form. And we have 4 stages in lysosome. As you know, primary, secondary, tertiary and autolysosome. So primary lysosome, as you know, it has inactive enzymes. So primary lysosome will have the inactive enzymes will be present. So it is not activated. Secondary will be having secondary will be having the vesicles with active enzymes. So the enzymes will get activated. Then we have tertiary lysosomes. See, this tertiary lysosomes will have the undigested particles. See, suppose you assume that we are eating a sambar rice. When we eat a sambar rice, we may eat all content, but some of you may throw off the curry leaf or pieces of vegetables, something, no? Like the thrown out content, which are undigested component, we can find it in tertiary lysosomes. Okay, so the undigested materials will be here. And of course, autolysosome. So only we are calling it a suicidal bag. So before we go into the autolysosome, can anybody tell me what is biological death? What is biological death, children? What is biological death? Anybody? See, when a person is dead, after some 20 to 25 hours, these lysosomes will be burst out and start doing phagocytosis of all cell contents. That time only we call it as autolysosome. So self-decaying, self-death of the cell is happening. That is why it is called autolysosomes. Okay, and these lysosomes generally we found in all cells except of course RBC and meristematic cells. Can you guess a cell where lysosomes will be more? Guess a cell where lysosomes will be more. You all know the answer, children. I know that you know the answer. Let me wait and see. Yes, Jayasurya, Sanjay, Ahmed, Shrikant, Jeevan, Nandika, Tiruvira, Nabita. Anybody? The lysosome will be more in that cell. Any guess? Where we have more lysosome. Okay, I'll give a clue. Shall I give a clue? Yes, Yogeshwari. The cell perform phagocytosis. Which cell? Now, I gave a hint. The cell will do phagocytosis. Can you guess, children? Any guess? 5, 4, 3. Any guess? 2, 1. Yes? Is it not WBC, my dear children? Where we have neutrophil and monocyte with phagocytic effector. So we can find lysosomes more there. Am I correct? Yes? Yes, children? Yes. Shall we proceed? The next one is vacuoles. Okay. So tell me where we have more vacuole, either in plant cell or animal cell. So vacuoles will be more in which cell? That is maximum occupied. Yes, Anishri. As you know, vacuoles are sac-like structure which holds cell sap. Of course, in plant it occupies 90% of the volume. Animal cell we have small vacuoles will be present. Yes, children, the newcomer can even answer me. If you know the answer, don't feel shy. We are all friends. Cell sap will be having what and what? Any guess, children? What is cell sap? What is cell sap, children? Yes. Yes, Balaji. Very good evening. Vanakkam. Of course. Okay. So plant will have the 90% volume, and of course animal is having smaller vacuole. Can anybody guess what is the composition of cell sap? Is the question. Any guess? Yes, Yogeshwari, Arini, Anamat, Sanjay, Tiruvira, Jeevan, anybody? Newcomer. It will be having in plant cell, of course, cell sap, water and sugar will be there. Okay, children. So, in case of the membrane, what that covers the plant cell is called tonoplast. So, it is what we call it as tonoplast. Okay. So, tonoplast is a membrane that covers the vacuole. And in case of animal cell, what are the vacuoles we have? We have contractile vacuole. What is the purpose of contractile vacuole? Any guess for any of you? Answer me. What is the purpose of contractile vacuole? I'll come back. Now we'll discuss about gas vacuoles. We already discussed it. And these gas vacuoles we found in cyanobacteria and also in purple green bacteria, photosynthetic bacteria. So we found. Of course, contractile vacuole is for osmoregulation. As you know, children. So it is mainly to balance water as well as electrolyte balance. Okay. Are you all clear? Yes, Sanjay. Agamath. Okay. So we finished all these. Let us continue the mitochondria. Mitochondria discovered by Kolliker. Of course, from insect wings. And where do we find mitochondria? So mitochondria will be more in the where energy is demanding. Okay. So in energy demanding cells, we find more mitochondria like the wing muscles of birds or insects. Okay. Muscles of humans. So where the energy is needed more, we have more mitochondria. Of course, mitochondria is not present in RBC as we know. Okay. And there are various number varies. Shape also varies. So it won't be the uniform shape. So here let us learn the structure of these mitochondria, children. So you can find the outer membrane. Can you see outer membrane? And also the inner membrane is found. This is the matrix of mitochondria and the foldings. So you can find the foldings of the mitochondria. These foldings are called cristae. And we find oxysomes in the cristae. Yes, asthma regulation and excretion is very correct. Excellent, Jeevan. And Narinishri, excellent answering. Yes. Yes, Purnima Prabhu. Very good evening. Very good evening. Happy to see your name. And we are discussing right now mitochondria. Okay. Now shall I continue? Any doubt so far? Any doubt, children? Shall we proceed? Yes, Nabita, you are very correct. Sugar and salt will be there in vacuoles. So except RBC, mitochondria found everywhere. Okay. Now let us continue. What are the functions of mitochondria? Can anybody guess the functions of mitochondria? So as we discuss the structure, it has outer membrane, it has inner membrane, there is intermembrane space, cristae, and on the cristae, of course, you can find oxysomes. So the oxysome has F0 and F1 particles. Yes. Any guess? What are the functions of mitochondria? Yes, it is what we call it as powerhouse. Very good. Excellent, Balaji and Ahmed. It produces ATP. Yes. Yes, very good, Jeevan. So it produces ATP is the correct answer. So only we call it as powerhouse of the cell. Yes, it provides energy production. Yes, Nabita, very correct. Excellent, 100% correct. Yes, Balaji, it provides energy. Is the correct answer. Yes, Nabita, very correct. It regulates metabolic activities. As you know, what are the process happens in the mitochondria? Krebs cycle is carried out in the mitochondria, and oxidative decarboxylation is carried out in mitochondria. So all these metabolism were regulated. Fine, very good, excellent. And 98% of ATP we synthesize here only, so only. Okay. And as you know, mitochondria apart from ATP synthesis, it also involved in vitellogenesis, yolk synthesis. So yolk synthesis, it plays a role. Yes, Nabita, metabolism we discussed. And it is also found in, yes, electron transport system. Is very correct, Ahmed. Excellent answering. Electron transport system, it is found. And then, any other? So the mitochondria which we find in sperm is called never-kind mitochondria. So it is called never-kind mitochondria. These are the points we need to remember regarding the mitochondria. Possible questions, sometimes labeling can also be asked, and where the Krebs cycle is carried out in the matrix. Intermembrane space, of course, electron transport system is happening. And where do we find the ATP production? Of course, F0 and F1. So these are the possible questions of mitochondria. Can I ask one more question, children? See, mitochondria is absent in bacteria. Who will carry out respiration in bacteria? There is no mitochondria in bacteria. Who will carry out respiration in bacteria? Any guess? Who will carry out respiration in bacteria in the absence of mitochondria? Yes, Nabita? Any guess? Yes, it is mesosome. 100% correct, Arinishri. Excellent. Yes, Tiruvira. Excellent answering. It is mesosome which will carry out, of course, the... Yes, Nabita, it is mesosome. The folding of plasma membrane in bacteria will carry out the respiration. Yes, Jeevan, 100% you are correct. Yes, Shalini. Excellent answer, Shalini. Yes, it is mesosome. Will carry out in the absence of mitochondria in bacteria the respiration. So next one we are going to discuss is about plastids. Of course, we can classify the plastids in three criteria. The colorful plastids are chromoplastids and green color plastids are the colorless plastids are leucoplastids. Okay. So under chromoplastid, of course, you have the chloroplast is coming. Leucoplast, let us discuss now. They are colorless. So they are mainly serving for storage. So they may store either protein or fat or starch. Any guess, children, which stores protein is called aleuroplastid. Alluro, how do we remember alluron layer in monocot seed if you remember the protein code. So alluroplastid. Fat we are storing in ilioplastid. So where we reserve oil and all that. Starch is called aminoplastid. Yes. So let us continue with the chromoplastid. It may be red in color or it may be yellow color. It may be green color also. As you know, green color we are seeing the outer membrane and inner membrane. Okay. And you find the sac-like structure is called what? Thylakoid. And you can find a group of thylakoid. Of course, we know it is grana. How one grana interconnected to another grana? What is the name of this? Any guess, children? One grana is connected to another grana by means of water. Yes, children. One grana connected to another grana by means of. Yes, Navita, excellent answering. It is stroma lamellae. Very correct. Excellent. 100% you are correct. Yes, Jeevan. Yes, Ahmed. It is stroma lamellae. Very correct answer. Excellent, Tiruvira. Yes, it is stroma lamellae. Where we find the connection. Yes, it is stroma lamellae. Yes, Arnishri, excellent answer. It is stroma lamellae which finds the grana and grana connection, of course. And the matrix we call it as stroma, where we do the photosynthetic work. Of course, you know, the light reactions are carried out where, children? The light reactions are happening in two places. Can anybody guess where and all light reaction happen in chloroplast? And where dark reaction will be carried out? Yes, Navita, very great. It is grana, the thylakoid place where we have the light reaction. Excellent. And stroma lamellae is the very correct answer. Yes, Tiruvira, excellent answering. 100% you are very correct. Yes, Mohammed. It is light reaction in thylakoid as well as in stroma lamellae. Yes, Vaishnavi. Very correct answer. Excellent. And dark reaction is carried out in stroma. Yes, this is about light and dark reaction. Yes, Shalini, very correct. Excellent answer, Shalini and Vaishnavi. Excellent answering. Yes. So light and dark reaction, you know, light reaction they prepare ATP and NADPH++, and dark reaction, of course, we prepare the sugar. Can anybody guess what sugar we synthesize during dark reaction? Is it glucose? Is it sucrose? Or is it starch? Or fructose? What sugar we synthesize during dark reaction? What sugar we synthesize during dark reaction? Is it glucose? Is it sucrose? Is it fructose or starch? It is glucose. Yes, Tiruvira and Jeevan, excellent answering. Yes, 100% you both are correct. Yes, Navita, excellent answering. Very correct answer. Navita, yes, Shalini, it is glucose. Actually, we prepare. Yes, Ravi Kumar, very good evening. Yes, it is glucose. Correct answer. Excellent, Ravi Kumar. Keep rocking. So let us continue. This is about chloroplast, of course. Possible question, as you know, if we store protein, if we store lipid or starch, what would be the answer? Yes. Are you all clear? So the next one we are going to discuss is about ribosomes. So who discovered ribosome? It is discovered by, answer me children, let me continue of course. You know, ribosomes are found in endoplasmic reticulum and in cytoplasm and in mitochondria, also in chloroplast. As you know, because of the presence of ribosome as well as DNA, they are considered to be semi-autonomous organelle both. Semi-autonomous organelle. Why? They have DNA and ribosome. So is it single strand DNA or double strand DNA, children? Yes, it is valid. Very correct. Excellent, Jeevan. Again, you're rocking today. Yes, it is George Pallet. Very correct answer. It involved in protein synthesis. Yes, Ravi Kumar. Very correct. Super. Yes, it is George Pallet. Is the very correct answer. Actually, fine. And can anybody guess what is the nature of DNA we have? Is it single strand DNA or double strand? Children? Yes, Arini. Very good evening. Yes, it is George Pallet, of course. It is single strand and you will be having C and G more so that it can withstand the temperature also. Yes, of course, let us continue. It is George Pallet. So only it is even otherwise called Pallet's particles. Yes, it is single strand. Very correct, Jeevan. Excellent answer. It is single strand with more C and G so that it can able to withstand the heat. Okay. So now let us continue the ribosome. So one possible question as we discussed, it is found endoplasmic reticulum, especially RER, and cytoplasm, mitochondria and chloroplast have the endoplasmic reticulum. The other question, which is called protein factory of the cell, as I showed you in the image, protein factory of the cell. Protein factory of the cell is dash. Yes. You wanted to see the image once again, children? Protein factory. So, which is called protein factory. Here you can see the factory and here you can see the protein. Yes, Jeevan, excellent. Excellent, Jeevan. 100% you are very correct. It is ribosome. Yes, Tiruvira. Yes, Arini. Yes, Shalini, Tiruvira and Navita, it is ribosome. Very correct answer. Excellent answer. Okay, so it is ribosome, which is the protein factory of the cell. So as you know, it involved in synthesis of protein. There are two types of ribosome. Yes, Ravi Kumar, very correct answer. Excellent, Ravi Kumar. There are two types. Can you guess what are the two types? It is 70S and 80S ribosome. So 70S is having 60% protein and remaining 40% is RNA. Whereas 80S is having 55% protein and remaining 45% is RNA. Yes, very good evening, Kavya. Very good evening. Hi, Kavya. Welcome you to the class. Yes. And you know, when many ribosome, yes, Ravi Kumar, excellent answer. It is 70S and 80S. When ribosomes are aligned, when many ribosomes are aligned, and involved in protein synthesis, it is called what? So I'll just recap the picture also for your reference. Can you guess what it is? When many ribosome joins and involved in protein synthesis, it is called what? Can you guess it, children? Yes, Jeevan, excellent Jeevan. It is polyribosome. Yes, Kavya and Dharini. Yes, Tiruvira, it is polyribosome. Yes, Navita, polyribosome or polysome is the correct answer. Yes, polyribosome or polysome is the correct answer. Excellent, Ahmed and Navita. Yes, Tiruvira and Jeevan, Dharini, everyone. Yes, polyribosome is the correct answer. So the possible question, as you know, children, where and all you can find ribosome and what are the distributions of the ribosome? Yes, Ravi Kumar, polyribosome is another possible question. Okay, children. And you know the parts of the ribosome, you have one small unit where mRNA will enter, of course, one big unit. The big unit having three compartments. One is A, which is the entry site. Another one is P, where is peptide bond is formed. Another one is E, which is the exit site. So the tRNA will leave off via this one lead. I hope you understood all about the ribosome. So cilia and flagella. So what are the difference between cilia and flagella? Cilia or flagella. Can you give an example of the organism which has cilia and which has flagella, children? So let me explain. Cilia usually will have one, flagella will be many, and they are oar-like structure with a synchronized movement. Here they are only one mostly. So this is regarding cilia. This is flagella. So usually there will be one. Of course, as you know, they are independent in action. Yes, excellent. Navita, paranecium cilia. Dinoflagellates and paramecium. Yes, dinoflagellate as flagella. Yes, excellent Ahmed and Nabita and Tiruvira, Arinishri. Excellent answering. Superb. So it is independent. As you know, children, the cilia will be more, they may be showing synchronized or metachronized movement. You can imagine a boat, when it is, when we row the boat, one after another. Yes, flagella sperm is also the correct answer. And Ravi Kumar, Euglena flagella is the correct answer. Excellent. So synchronized and metachronized movement you can see on the cilia. What is the structural and functional unit of cilia and flagella? What is the structural and functional unit of cilia and flagella, children? The structural and functional unit. Yes, Shubh Agnihotri. Very good evening. A warm welcome to you. What is your doubt? Functional and structural unit. Axoneme. Axoneme is very correct answer. Excellent answer. Is the correct answer. Axoneme is Vaishnavi. Very correct answer. Okay. So you can find the cilia and flagella is the correct answer. Okay. And you can see here the 9 will be on the periphery. So the 9 will be on the periphery and 2 will be in the center. Yes, axoneme is very correct answer. Excellent. We are doing class. Kindly don't disturb us. 2 in the center, which is we call it as hub. So others are learning. If you are not in a mood to learn, please you can enjoy it separately. So the 9 will be in the periphery, 2 will be in the center, which we call it as hub. Can you tell me, children? So the axonemal arrangement 9 plus 2 will be for which organism and 9 plus 0 will be for which organism? Yes. Yes. So 9 plus 2 will be the arrangement. 9 plus 0 for prokaryote. Very correct answer, Tiruvira. Excellent answer. Very good. You are rocking today. Yes. It is 9 plus 2 eukaryotic organism and 9 plus 0 is for prokaryotic organism. Okay. So cilia and flagella, of course. So it is helpful for locomotion. Other difference you can see cilia is useful for locomotion and also you know about stereocilia, children. What is the purpose of stereocilia? Can anybody answer me? What is the function of stereocilia? Can anybody answer me? What is the function of stereocilia? Of course, flagella is only used for locomotion. Can anybody answer me? Where do we find stereocilia? Yes, Nabita. Harini. Yes, Tiruvira. Excellent answering. Excellent. You are rocking. Yes, it is sensory cilia. Very correct. Excellent. Yes, it is sensory cilia. Can anybody tell me where do we find the stereocilia? Where do we find the stereocilia? Yes, it is on sensory cell. Excellent, Agamal. Yes, Nabita. For hearing as well as yes, Yogeshwari. Yes, Agamal. Excellent answering. Yes, it is for sensory. We could find it in the organ of Corti. Of course, yes, organ of Corti. Yes, Jeevan. Very good evening. Yes, correct answer. Very good. Excellent. Yes, Tiruvira. Organ of Corti we find even in the glasopharyngeal, that is in the tongue, to analyze the taste. You may have learned, children, like when we have sensory papilla, we have the nerve, so it may dissolve. Of course, the nerve will create the depolarization, and we can able to understand the taste of the food with the help of this stereocilia. So sensory function is the very correct answer. Excellent. Dharini, Tiruvira, Jeevan, Navita, excellent answering. Okay. And the flagella is only performing locomotion. So it is not performing any sensory activity. Other than that, types. So the cilia, which is helpful, the types, of course, we discussed stereocilia as well as kinetocilia. And flagella is of two types. Of course, one is whiplash flagella, another one is tinsel flagella. One is whiplash flagella, another one is tinsel flagella. This is regarding the flagella. I hope you are all understood. From where cilia and flagella originate, children? From where cilia and flagella originate? From where cilia and flagella originate? Anybody, can you answer? Can anybody answer from where it is originating? Is it from? Yes, it is from basal membrane. Yes, basal body. Yes, Jeevan. Yes, centriole or centrosome, which also originate from the basal body, is a very correct answer. Basal body. And these central one, when it rotates only, the peripheral ones will also rotate. Can you name some of the protein, children, which are found in the cilia and flagella? Yes, microtubular organizing region is basal body. Very correct answer, Tiruvira. Excellent. You're rocking today. Yes. Can you name protein, children? What are the proteins we generally find in the cilia and flagella? So the proteins are tubulin, dynein, nexone. So these are the proteins we can generally find in the, of course, in the microtubular structure, that is in the cilia and flagella axonemal arrangement. Centrioles. Yes, excellent, Tiruvira. Yes, tubulin is the correct answer. Centriole. The arrangement of axoneme is 9 plus 0. Centrioles are found exclusively in animal cell. True or false, children? Centriole are found exclusively in animal cell. Is it true or false? Is it true or false? Centrioles are found exclusively in animal cell. Yes, true is the very correct answer. Jeevan, you're rocking today. Jeevan, excellent answering. Yes, Ahmed, Tiruvira, excellent. Yes, Dharani, 100% you're correct. It is true. So we found only in animal cell. What is the arrangement? 9 plus 0. If a pair of centriole is found, it is called what? Yes, Navita and Arnishri. A pair of centriole is found. It is called a pair of centriole is found means it is called dash. Yes, Vaishnavi, very correct answer. It is true. Navita, it is correct. You're all rocking today. Girls, a pair of centriole is found. It is called. Yes, children. Any guess? Yes, it is centrosome. Yes, Tiruvira, very correct answer. Excellent. Yes, a pair of centriole is called centrosome. Yes, it is centrosome. As you know, it play a role in cell division. The arrangement of centrosome is called the cartwheel arrangement. Cartwheel arrangement is a possible question, children. As you know, 9 you will find, it will be triplets actually. 9 in the periphery like this and 2 in the 0 in the middle actually. You do not find any central one. So only it is 9 plus 0 arrangement in centriole. Yes, Yogeshwari. Yes, it is centrosome. Yes, Ravikumar. Very correct. Where you went in the middle? Yes, it is centrosome. Very correct answer. 9 plus 0. Very correct answer. Excellent, children. So I hope you understood centriole and centrosome. And who is the gang leader of the cell? The gang leader of cell is okay. So I am waiting for your answer, children. Only yes, yes. Nucleus is very correct answer. Yes, Nabita and Jeevan. Yes, Vaishnavi and Dharani. Yes, the gang leader of the cell. Okay. Or who is brain of the cell? Of course, it is nucleus. Who discovered a nucleus? Yes, Yogeshwari. Very correct answer. Excellent answering, Yogeshwari. Who discovered a nucleus? Yes, it is Robert Brown. Excellent answering. Yes, Jeevan and Nabita, both of you are rocking today. It is Robert Brown. I hope you remember the Brownian movement even. Yes, Ahmed. Yes, Tiruvira, Arinishri. Excellent answering. It is Robert Brown. And what is the structure of nucleus? So you can find a nucleolus here, of course. Does a nucleolus has membrane or non-membrane organelle it is? Can anybody answer me? The nucleolus is a membranous or non-membranous organelle? Is it a membranous or non-membranous organelle nucleolus? Yes, you can keep answering. Meanwhile, we'll discuss. Of course, nucleus will have the nuclear membrane. So you can see it is a double membrane with a lipid and protein. Okay. And of course, so lipid and protein is there. It is a double membrane layer and you can find, of course, a gap between the layer. The gap is called the nuclear pore. So it will allow only certain things into the cell, certain things outside of the cell. So just like a plasma membrane, the nuclear membrane also serves for us. Okay. And then of course nucleolus, I asked the children, can anybody answer me? Is it having membrane or non-membrane? I asked. Of course nucleolus is a non-membrane bound organelle. Okay. So it is a non-membrane bound organelle, children, nucleolus. Yes, Arvishri. Yes, Nabita. Yes, Ravikumar. And yes, Ahmed. Shalini, Tiruvira. Yes, it is non-membrane bound organelle is 100% correct. You were all rocking. Excellent. Yes, Kumari, it is non-membrane. Very correct answer. Excellent. And Jeevan, non-membrane bound organelle. And you know, RNA are formed here, especially rRNA, tRNA are formed here. And also satellite chromosome are formed here only. Okay, so this is regarding the nucleolus. Then of course, about the chromatin. So we have nucleoplasm, whatsoever it is around, you know, it is nucleoplasm, where we find enzymes, ATP, okay. So we find enzymes and we find ATPs, all these in nucleoplasm and also chromatin. So let us see the difference between chromatin, chromosome, chromatid. So the chromatin, you can find the diffusion of fiber-like structure in the nucleus is what? Chromatin. So the diffused fiber-like structure in the nucleoplasm. Okay. The condensed form of chromatin, we call it as chromosome. Yes, sir? Am I correct, children? Yes. So it is the condensed form. Of course, it is what we call it as chromosome. And chromatid, as you know, when the DNA, yes, hair-like structure is chromatin. Very correct. Excellent, Ravi Kumar. So it is diffused fiber-like structure. The condensed form of chromatin is chromosome. As you know, in S phase, the DNA doubles. When the DNA doubles, it is what we call it as chromatid. So we used to draw like structures like this, na? Chromatid. So this is chromatid. Are you clear, children? Understood? So what are the functions of nucleus? What are the functions of nucleus? Any guess? Hi Tirumala, very good evening. Can you list out what are the functions of nucleus? Yes, it involved in RNA synthesis. Excellent answer, Ahmed. RNA synthesis. As it is having the chromosome heredity, information are passed, and chromosome is there. It controls all other activity. DNA is there. Control the other cell organelle. So only it is brain of other cells. Brain of the cell organelle. Other cells. Yes. Any other function? Yes, it also involved in protein synthesis. 100% correct, Jeevan. Yes, Tiruvira, rRNA synthesis. Very correct. Excellent. Any other answer? So it also involved in protein synthesis. And it holds enzymes. As we know, endonuclease, exonuclease, ligase, polymerase, all these we find. And we also have some minerals like manganese, magnesium. So all these minerals also we found in the nucleus. Okay, so their abundancy and reduce level may decide the translations, other process of the cellular activity. So we are proceeding to the chromosome. Yes, DNA replication, transcription, processing are carried out in the nucleus. Very correct, Tirumala. Excellent. Excellent. You're rocking. Excellent answer. DNA replication is carried out. Cell division is under the control of nucleus. Yes. Fine. Chromosome. Who coined the term chromatid? Yes, cell division. Very correct answer. Chromatid. Who coined the term? The term chromatid coined by, start with the name F. Yes, it controls the enzyme and protein synthesis. Yes, Tirumala, very correct. Excellent answering of nucleus. Of course, chromatid is coined by, any guess? Yes, it is Fleming. Very correct, Jeevan. It's your day, Jeevan. Fleming. Yes, Navita. Fleming only coined the term chromatid. And of course, chromosome, if you take, you have two arms. So, the two arms are here. And this is called centromere or primary constriction. And surrounding the centromere, you know, it is called kinetoblast, where only the spindle fiber will attach. Okay. And you know, one arm will be long and other arm will be short. P arm and Q arm. In detail, if you proceed, of course. And these chromosomes only carry the DNA inside. We have the DNA, of course, which carry the hereditary material. You know, the nucleosomes and all. Histone proteins are there. These are the parts of chromosomes or types of chromosome. When we have equal arms, it is called what? Yes, Yogeshwari, it is very correct answer. Fleming, no doubt. It is Fleming. Yes, Tirumala. we are storing the RNA, DNA, etc. in, of course, nucleus. So equal arms when we have, it is called what? When we have equal side arms. Yes, Fleming is very correct. Yes, Navita, 100% correct. It is metacentric. One long arm and one short arm. Then it is called one longer arm, one shorter arm. One longer arm and one shorter arm is called yes, Navita, excellent answering. Submetacentric. Yes, very correct, Navita, submetacentric. One very long arm and one very short arm is termed to be yes, Ravi Kumar. Yes, Jeevan, excellent. Yes, Kumari and Tiruvira, submetacentric is the correct answer. Excellent. Excellent answering. Yes, it is acrocentric. Very correct. Navita again, excellent. Yes, acrocentric is the correct answer. Ishwari as well as Kumari. Yes, Jeevan. And if you have centromere in the terminal arm, so centromere in terminal position, it is called what? Centromere in terminal position. Centromere in terminal position. Yes, Navita, it is telocentric. Very correct. Excellent answering, Navita. Excellent. Yes, Kumari, it is correct answer. It is telocentric. Yes, Ravi Kumar. It was telocentric actually when you have centromere at the distal end. It is telocentric. Yes, Jeevan, very correct answering. Excellent answering by all of you. Yes, it is telocentric. And certain chromosome you can find secondary constriction, and the secondary constriction is otherwise called SAT chromosome. So SAT chromosome is a secondary constriction, of course. This SAT chromosome will be involving in the process of, as you know, polymorphism. Individuality of ours is responsible because of the SAT chromosome. We will be learning in 12th standard portion, of course, about SAT chromosome in fingerprinting. The satellite DNA is involved in the identification and all that. So that is the role of SAT chromosome. I hope you all understood. Microbodies. Yes. Microbodies are spirosomes. So these spirosomes are otherwise called plant lysosomes. So they have hydrolytic enzyme like protease, lipase, carbohydrates, alkaline phosphatase. Alkaline phosphatase is a marker enzyme. So alkaline phosphatase is a marker enzyme. It is exclusively found in plant. They may ask you the question, The plant, where do we find? Of course, only in plant. It is spirosomes, of course. Then we have glyoxysome. Glyoxysome is for beta oxidation, where we convert fats into glucose. So as you know children, when we were starving, we didn't eat means what happens? Of course, the energy we are gaining from fat only, next source after glucose. Fat will be converted into glucose and we gain energy. Okay, fine. Then after the fat, of course, protein will also provide us energy. This is glyoxysome. These two are found exclusively in plant. Peroxisome. Found both in plant as well as animals. Of course, it is for removal of toxins. So any amino acids when we digest, I am writing AA for amino acid. So when we treat the amino acid with catalase, you know, some hydrogen peroxide will form, H2O2. See, there is a funny line also. When we drink water, we survive, we dance. When we drink H2O2, somebody else will dance in front of us. So, person cannot survive. It is a poison. H2O2. Peroxidase will process and form water. Of course, oxygen. So, this is the role of peroxisome found in both plant and animal. Only microbody with the plant and animal is peroxisomes. Okay. I hope you all understood the class. Any doubt children, you can ask me. Let me share you a small incident. You all know about Mahabharatam story. So மகாபாரதம்ல உங்களுக்கு நல்லா தெரியும். அதுல வந்து பாண்டவாஸ் அண்ட் கௌரவாஸ் ரெண்டு டீமா இருந்திருப்பாங்க. ஆனா அவங்களோட குரு பாத்தீங்கன்னா துரோணாச்சாரியா அப்படின்னு ஒரே ஒரு குரு தான் இருந்தாரு. அவர் தான் எல்லாமே சொல்லி கொடுத்தாரு. அப்கோர்ஸ் இந்த ஆர்ச்சரி, அதாவது வில் வித்தைன்னு சொல்லுவோம் இல்லையா தமிழ்ல, அந்த ஆர்ச்சரி அப்படின்றத சொல்லி கொடுக்கும் போது அவர் எல்லாருக்கும் ஒரே மாதிரி தான் சொல்லி கொடுத்தாரு. அப்கோர்ஸ் அதுல வந்து எக்ஸ்பர்ட் யாருன்னா அர்ஜுன். அவர் தான் ரொம்ப எக்ஸ்பர்ட்டா இருந்தார். ஒரு நாள் என்ன பண்றாங்க, வை ஆர் ஆல்வேஸ் பிரைசிங் அர்ஜுன் அப்படின்னு சொல்லி மற்ற ஸ்டூடெண்ட்ஸ் எல்லாம் கேக்குறாங்க. டீச்சர்ஸ் பெட் அர்ஜுனா இருக்கிறாரு அப்படின்னு. அப்போ அவர் என்ன பண்றாரு, எல்லாரையும் நிக்க வச்சு ஒரு மரத்தை போக்கஸ் பண்ண சொல்றாரு. அதுல ஒரு பறவை இருக்கு. அப்கோர்ஸ் அவர் கேக்குறாரு, உங்க எல்லாருக்கும் என்ன தெரியறது அப்படின்னு. அப்போ உங்களுக்கு தெரியும், the participants of the மகாபாரதம் இல்லையா, தர்மர் சொல்வாரு, எனக்கு எல்லாம் தெரியறது, அப்கோர்ஸ் கிளி கூட தெரியுது அப்படின்னு சொல்றாரு. அப்ப வந்து பீமா எல்லாரும் என்ன சொல்றாங்கன்னா, எனக்கு வந்து மரம் தெரியறது, அதோட கிளை தெரியறது, இந்த பறவையோட கால் தெரியறது அப்படின்னு எல்லாரும் ஒருத்தர் ஒன்னொன்னு சொல்லிட்டே வர்றாங்க. அப்புறம் நீங்க பாத்தீங்கன்னா அர்ஜுனோட டர்ன் வருது. அப்ப வந்து குரு கேக்குறாரு, அர்ஜுன் உன்னால என்ன பார்க்க முடியாது அப்படின்னு கேக்குறாரு. அப்ப அர்ஜுன் சொல்றாரு, எனக்கு பறவையோட கண்ணை தவிர வேற எதுலயுமே தெரியல அப்படின்னு சொல்றாரு. இது ஏன் அப்படின்னா, நம்ம படிக்கும் போது நிறைய டிஸ்ட்ராக்ஷன்ஸ் வரும். இப்ப நம்ம கிளாஸ்ல கூட கொஞ்சம் டிஸ்ட்ராக்ஷன் இருந்தது. அந்த மாதிரி நிறைய டிஸ்ட்ராக்ஷன்ஸ் வரும். அப்படி வரும்போது நம்மளோட ஃபோக்கஸ் என்ன அப்படின்னு பாத்தீங்கன்னா, படிக்கிறது மட்டும்தான். ஸ்டெத்தோஸ்கோப் வச்சுக்கணும், ஒயிட் கோட் போடணும். அந்த ஒயிட் கோட் நம்மளோட லெஃப்ட் ஹேண்ட்ல அழகா தொங்க விட்டுருக்கணும், அலாங் வித் ஸ்டெத்தோஸ்கோப். அதுதான் நம்மளோட எய்மா இருக்கு. அதுக்குதான் நம்ம கஷ்டப்படுறோம். அதை வந்து டீவியேட் பண்ணாம வேற எதுலயுமே செதறாம, லைக் ஹவ் அர்ஜுன் கேன் ஏபிள் டு see only eye of the parrot, அதே மாதிரி உங்களோட ஃபோக்கஸும் இருந்ததுன்னா கண்டிப்பா நம்ம சக்சஸ் ஆகும். ஆல் தி பெஸ்ட் சில்ட்ரன் ஃபார் ஆல் ஆஃப் யூ. ஐ ஹோப் யூ ஆல் அண்டர்ஸ்டுட். எனி டவுட் யூ ப்ளீஸ் ஆஸ்க் மீ. இந்த டிஃபரன்சஸ் உங்க எல்லாருக்கும் தெரிஞ்சதுதான், ப்ரோகாரியோட்டிக் அண்ட் யூகாரியோட்டிக் செல்ஸ். கேன் யூ கெஸ் which is the dustbin of the cell? Yes. I hope you all enjoyed the class. எல்லாருக்கும் பிடிச்சிருக்கும்னு நினைக்கிறேன். Class புரிஞ்சிருக்கும்னு நினைக்கிறேன். Don't forget to give your likes also and comments also. Thank you all for participating. Thank you everyone. My heartfelt gratitude and all the best, children. Any doubt, please tell me. I'm waiting for your reply. Yes, it is vacuole. It is vacuole. Very correct answer, Ahmed. Excellent answering, Dharani. Yes, Jeevan and Shalini. Yes, Tiruvira. Very correct answer. It is vacuole. So be focused on your studies. இந்த ஏஜ்ல உங்களுக்கு நல்லா தெரியும் நீங்க எல்லாம் சயின்ஸ் ஸ்டூடெண்ட்ஸ் ஹார்மோன்ஸ் நிறைய செக்ரீட் ஆகும் நமக்கு டீவியேஷன் இருக்கும் கொஞ்ச நேரம் வாட்ஸ்அப் பார்க்கலாம்னு தோணும் அந்த கொஞ்ச நேரம் ரொம்ப நேரமா மாறிடும் ஸோ ஃபோக்கஸ் ஒன்லி ஆன் யுவர் எய்ம் ஆம்பிஷன் white coat as well as stethoscope. Yes. Yes, vacuole is very correct. Yes, craft rainbow. Very correct answer. Excellent. Your Yogeshwari. Very correct. Jeevan. Yes, thank you, Jeevan. Thank you so much. You all rock today, children. Excellent performance for everyone. Keep rocking like this. Thank you all for your participation. Thank you, Nabita, for your words. Thank you. Please put it in the comments so that many other children will also come. Give your comment, give your likes. Thank you, Nabita. Thank you. Thank you, Yogeshwari. Thank you, Tiruvira.